Welcome to MOOC course on Introduction to Proteogenomics. Today we have scientist Dr. Suman Thakur from Center for Cellular and Molecular Biology, CCMB Hyderabad. Dr. Thakur will talk to you about mass spectrometry based quantitative proteomics and how it can help the cancer research. He will also talk about why quantitative proteomics is very important, in which way various tools available for doing quantitative proteomics have helped to understand different diseases. Dr. Thakur will focus on why we are not getting clinically relevant biomarkers for all the cancers and why one drug cannot cure all the cancers. He will talk about different specifications for mass spectrometry based columns, for example, the gradients, the column length and other type of mass spectrometry parameters, how it could be optimized to obtain good results. He will also talk about why the number of proteins decreases in labeled analysis as compared to the label free quantitative analysis. He will then talk about anti-cancer compound screening and how the proteomics, cell biology and animal studies together could only help to find out the clinically relevant information. So, let us welcome Dr. Suman Thakur for today's lecture.
Mass spectrometry based quantitative proteomics in cancer. So, anyhow, you are well trained and thanks to all the previous speaker who has set the stage, and I should just move it. So, all you know proteogenomics. So, genomics is here, proteomics is here, middle transcriptomics, metabolomics. For us these all are different subject, but for body all are happening. Billion year, trillion year of evolution has made this. So, this is not a one day anything, maybe this is new subject for us. But what is here? So, biology change, 1000 years before also question what is life, today is also question what is life, most probably 1000 years later will be also question what is life, what is evolution. But what changed, that you have to understand, technology. So, technology drives biology. You get new technology in your hand and you try to answer different question. If you want to do something in life, you have to ask question. What is the question? Find a specific anti-cancer compound for particular cancer without side effects. Is it possible to find a drug without side effect? That is the reason you ask question, what is not easily possible. Second, find earlier stage and cancer biomarker. Cancer is such a word, till you discuss in symposium and lecture hall it is fine. If you see any patient, your near and dear, you will be shaken very badly. So, that is a bad word, is a cancer basically is a disease. Still after so many years, 100 years of research, we are not able to cure it. We are still in the middle. So, what you need? Early stage. Every doctor asks early stage. But what is the question? Common cause. There are different organs in the body, different types of cancer are there. If all are there, then cancer should be common. This question you can ask, and we try to ask this question, and I thought select 11 or 10 cancer cell line. It is very nice to, we have to get clear, this is the cell line. Same thing we are trying to repeat on the tissue also. So, you take HeLa, cervical cancer, Jurgut, leukemia, HepG2 liver cancer, GAMG brain tumor, MCF breast cancer, A549 lung cancer, LNCP prostate, RKO colon, U2S bone and K562 again myeloid. So, different type of cancer are there. Why not one drug is there for all the cancer? Impossible. Origin is different, metastasis is different. So, that means it is very complex. And then you have to take, if you want to do anything in the proteomics, you have to select one control. This is non-cancerous cell line. So, we thought we will take this 10 cancerous cell line and very childish way you think, you get 5000 proteins, so then in L10 or 11 you will have almost whole human proteome. Practically it is not possible. Or you do different fractionation and you come to 10,000 protein in each cell line, still you will not able to cross 11,000, 12,000, maximum 13,000. So, what is the matter that means? All proteins in all the cancer are same, only quantitation is getting changed. Some place someone is expressed, some place someone is expressed. So, that means what? Expression level, quantitation. So, finally what came? Quantitation is important in the experimental, and it is happening inside the body. But how you will do this? This is very simple. We want cancer full proteome, that is one or few proteins, protein chemistry. You will see some lab is whole 20 years has been focused on one protein, but some lab will not like one protein. Why one protein? We should have 1000 protein. But what you will do with 1000 proteins? Again, what's miss? Whatever is your interest that you have missed. So finally, if you want to make a drug or target, you will target on one protein. So what proteomics is giving you? Completely back cycle to find the target and come back on the one protein and make the drug. That's same things we are trying also to do. But before that, I will go with little bit problem is there. There is nothing, everything is good in life. But mostly we talk good things. If you go to search the biomarker things, different disease, same marker. Same disease, different marker. All you will get confused and you will end up with the nothing. So bottom line, what we need unique biomarker. That's the whole thing, and field should grow it. Or maybe you need set of biomarker. Just like if someone is telling, no, this is this disease, then again you go to cross check, you go to verification. That thing has to be done. Why all these things is happening? Answer is here, billion year of evolution, you can't easily search, find in one day, directly sort it. It has to be systematic a study, or if you are by luck, you are hitting in the dark, it got hit. Both are happening together. And both example I will show you how we fail, how we pass. We thought instead of 15 centimeter, make half a meter column. Think little bit weird, everyone will tell you crazy, it works, you are best. If did not works, you still carry with the tag crazy. Okay, so now 15 centimeter, 50 centimeter column, it's long column, make bit side everyone using 5 micrometer, 3 micrometer, half. What will happen? HPLC pressure will very increase. To increase the pressure, simple use school knowledge, Charles law, Boyle's law, heat it, down the temperature, increase the temperature, down the pressure, and somehow you will manage with old HPLC, this 50 centimeter column, long run, and 5000 protein came that time in one single shot. This was just example. Same technique we have developed. Now recently we have published how long gradient we can use, and after 12 hours if you are using the gradient, recently published in 2018, there is no use. So it is saturated. So need of fixation, how long gradient, how long this and that. Now this technique you should use in Silek. That I am going to tell about the cancerous cell. So this is just introduction. Now I show you label free quantitation, 5000 protein comes in one run, but when you do Silek, you get less protein. Why? Because now 2 peak is needed to identify and quantify a protein. So you increase the complexity. If you increase the complexity, sensitivity has to be compromised. Okay? Or your protein number has to be compromised. But we know all the way how to increase the number also. What? If in 1 hour you got 3700, make triplicate, no? And then quantify. Here bioinformatics, you like or dislike bioinformatics, you have to use. Okay? So now this is the place, 1 into 8 hour you got 3000, now run triplicate, you get 4222, because which one peak has come in this run, another peak came in this run. By bioinformatics you merge and you can increase that. So this way you are showing. But there is the way, triplicate, 4, 5 times, 6 times, how many times? This is showing almost in triplicate on 4, almost you are getting close. So there is no need of going to unnecessary too much. Okay? You have to keep your temptation and limitation. That is also very important. Otherwise you are doing only one thing. Now same thing we did with all this 11 cell line. Okay? And then we try to find, is there any common thing is there? Or we have to stop on 11, first we did 1, then gone 5, then 11, now we are planning to have 25 or 50 cell line to get complete idea. But we can't decide in one, it's a very costly affair, and we have selected in the broad range. Now by this, if you do deep proteome, you will get 10,000 protein in each cell line by fractionation. But we thought we should go single shot also, and we should see in one run. There is no comparison between any fractionation, 4 fractionation, unite, no bioinformatics, one run, direct result and see. That both approach we have used. Now you see what happens here, it is single shot. one shot whatever came. This is CID, Silect, you know CID, HCD, you would have heard, okay. High energy, today is the era of HCD, okay. Now, you take HAC 293, light plus heavy, okay. So, HAC is there, light and heavy. Now, you are comparing that, there should be theoretically any difference in quantitation, upregulation, downregulation? It should not. That is the reason when you quantify, identify 4200, I quantified 91% in 4. But when you did in 1, I told you already concept, here you quantified only 78%, here 89%, here 91%. So, what happens when you do multiple run, you are going to quantify more. But now I am increasing 5 cell line, I am mixing 5 cell line together and then I am trying to see what is happening. One thing you see, if one cell line gave me 4299 protein, mine tells 5 cell line will give more. Correct or not? Simple thinking, do not think too much. Answer is no. Then we thought 11 cell line will give more. Answer is again no. See, this pattern is containing single run is giving less, triplicate is giving more and here you are getting more. But when you are increasing the cell line, nothing is changing. That shows your technology has limitation. Of course, 5 cell line when you put you have more protein, 11 cell line when you put you have more protein, but technology has limitation. That means still our so called well developed, well costly mass spec need to develop less or more? Very more. So, still price will increase, development will continue. Now, see, now this is same CID, here compare HAC 293 versus HAC 293. So, there is theoretically there is no difference, okay. When no difference, so what happened? Anyhow in quantitation we do not take less than 2-fold, okay. See, majority of things is falling in this less than 2-fold and whatever it is showing 1%, that is almost a error, okay. Now, I have taken 5 cell line, in 5 cell line I am taking HeLa versus Jurgat, HeLa, HapG2, GAMG, FCF7. So, this HeLa is inside that 5 cell line, but I am able to quantify and see 2-fold, that is falling only 84%, that means others are changing, okay. Here is the very less change, here you can see more change, that means different cell line has different things, correct. Now, here I took 11 cell line, it is coming almost same. That means because my technology is also coming close and result is also coming close. So, I can't comment on that, clear. But that shows me 5 cell line has more protein or different protein compared to that, and if theoretically same 2 cell line if you compare, where is upregulation, downregulation? If here there you get upregulation, downregulation, that means better to stop the experiment, okay. So, that shows your control, things are going in right direction. But this HeLa is inside the 5 cell line and 11 cell line. With this you plot, you have learned all the R and all these things today. By plot you can see that 5 cell line is not that much sharp, 11 cell line is more that much sharp. That means quantitation is better when you have multiple cell line, and chances to find that protein is very high. Same things you do with HCD, 1, 5, 11 cell line, and you will get different type of things, and we are fine here. But this HeLa is again inside that. Now, HeLa compare with Jurgat, you will get only 70%, no changes almost. But HeLa with 5 cell line, HeLa with 11 cell line, you are getting more to cover it. Now, it is different you are comparing. Again you see that HeLa with Jurgat, your line, your graph is not density plot is not so sharp. Here it is better and 11 cell line better. So, this is giving me indication that my 11 cell line or higher stock, I will able to cover more protein of that. If that mix is ready, that proper things is ready, that means I will have more chance to quantify, because in human I can't label select, okay. It is only possible in cell culture, okay. Now, you see here we have taken 1 cell line HCC 1599, breast cancer. This is not mix present in the mix of 5 and 11. Now, you see here chances of getting quantification is more or less, because it is not inside, you will get different things. So, this concept is coming clearly how you make the mix, do you need a master mix, do you need a this, and whole analysis idea still we are doing, that what common protein is there in all the type of cancer, where it is getting click or where it is getting a start, that is still under the way. Most probably next time I will show you more and better. Same things I am trying also to go with the tissue now. When 11 cell line we can take, now we know what is going to happen. Now 11 tissue or 20 tissue we are trying to do, all cancerous tissue, and we are looking what is the common, and can we find one common place where all this get triggered. That will help. We do not know what will happen. With this, you again see this density plot, always 11 cell line is coming better. Okay? You all have learned R, and with this I will move towards the little bit cancer drug discovery. That is my favorite hard work of PhD student, and it is somehow giving good result. So, what is there? Development of anti-cancer compound, use cell biology, mouse model and human model. You have to go finally clinical trial. So, that is the what simple. Cancer means tumor, break the cluster, induce cell death, apoptosis, and reduce cancer cell proliferation. Three things as a target, different compound we have a screen. There are, you would have heard a company screen 10,000, 15,000 compound library and then come to one target. We thought how can we do a smartly. Here this is the childhood cancer, mostly happen in childhood time before 5 years. Why it happens? It is very tough to tell. No one knows. It can happen in one eye, it can happen in two eye, and both the eye can be affected. There is the drug with very high side effect, carboplatin, etoposide and vincristine, mostly natural towards. Okay? So, we thought nature maybe have something. With this, now how to think about the cancer, how much. Unfortunately, this retinoblastoma is very high in India or developing country, India, Africa and all this. Why? There is no answer of that. But if you look all these things, you will see 10% of pediatric patients have retinoblastoma. Okay? In that also, highest incidence of retinoblastoma is in Africa and India. Why? No one knows. This is the cancer related with the gene, because you are in proteogenomics. And this cancer is related with the gene retinoblastoma RB. You would have heard cancer suppressor gene, and this is directly linked with that. Okay? So, this is sometime hereditary, sometime environmental, but there sometime no one understand why it happened. Okay? Now, people are still thinking which place it is getting a start, and recently one in 2014 paper came that they tell it is mostly starting with the cone precursor cell. Okay? So, this is the little bit evidence has come. So, this is below 14 years age, non-hereditary is 60%, hereditary is 40%. So, wonderful model system to study the cancer where genes, genetics is also involved, environment is also involved, but no one has any answer. Okay? Now, this is terrible, this is not good to see too much. Okay? In 6 months times, this become terrible. Okay? If you did not get treatment, then very tough. That is the problem happen in developing country. When you see leukokaria, you can characterize, if a doctor will see a normal person will quickly figure out there is something in the eye. This is the step where you understand that there is something, leukokaria step you will tell. I have mouse, retinoblastoma mouse in lab, we see that directly it. Okay? But if it get bigger, if they get tumor bigger than head size. Now you can understand how this happens. If timely there is nothing, enucleation is only that, a chances to having metastasis is very, very high. So what you should do? Now this is the chain, and most of the time what happen, children, parents not able to take to the hospital, no one takes care, it gets aggressive stage, then it is almost nothing. Now, so what we need? We need a need for the development of safe, better and effective manner. What you need at this time? You need some drug. Okay? Whatever drug is there, that is also not able to cure, it has very side effect. Then this, one of the student, Kamakshi, PhD student, has done this work. How? Now we thought to break the cluster. Okay? And now idea is that find a drug and find mode of action. In science, mode of action is more important. Drug needs patient, mode of action will give you patient help and to make the another drug better. Okay? So, now simple concept, break the cluster. You see this is cluster forming cell. Okay? So, now when you have started giving this control, cluster is there. You give, we have screens few compounds, several compounds, and then we came to this compound, this break the cluster. But this is not a big deal. You put surf excel, will, anything, it will break the cluster. So, there is nothing conclusive, but indication is there. Okay. Now, if you got what a biologist will do, quickly go and do cytotoxicity. Okay. So, we did cytotoxicity and somehow we found Y79 cancer, retinoblastoma cancer cell line is giving 18 micromolar IC50. But if you got 18 micromolar in the cancerous cell, it is also possible this will kill your normal cell. Okay. If it is killing normal cell, then how can this will be drug? First argument you ask yourself and you confirm here. Now what we did? We took ARP19, it is also retinal epithelial cell, and here you see IC50 is 165 micromolar. That means there is, it is not killing normal cell. We are looking for this. We are looking a compound which has potential to kill only cancerous cell, not to normal cell. That is the whole point came here. So, now you see this is our slide. Anyhow, there is nothing hard to tell fact. This experiment we did very late. First we did because we do not have cell line, we tried to arrange cell line, it took a lot of time. Later we did, but somehow it worked. Now, here proteomics played the role. Now, I told you, you put surf excel also it will work. Okay. But when surf excel concentration will go, it will come again cell closure. Okay. Now, in this compound, this never comes close. So, that was one benefit. Now, you took the, give the treatment with this compound to different cell, okay, both, and now you do proteomics. I have shown you how to do it. Okay. Now, you do proteomics and now you start comparing what happened after doing proteomics. When you look the proteomics, what you found, cell adhesion related proteins are down regulated in E4. E4 is nothing, it is my lab number, room number. Okay. So, that is from it came. So, now all cell adhesion related protein, so cell is not getting attached, it is getting detached and cell adhesion protein is getting down regulated, that means something is going towards the cell adhesion. No one is going to believe proteomics if you are not doing antibody or some validation, at least reviewer. Okay. We all believe. Okay. Reviewer will ask you. Now, we have done antibody experiment and we have moved forward. Now, if it is really anti-cancer, all oncogene and cancer related proteins should be down regulated, perfectly hit. Okay. So, that means it is anti-cancer. Now, you have to talk about the mechanism. What happens if all mitochondrial protein you will see that is getting upregulated in the treatment. So, mechanism is somewhere related to mitochondria. Who is telling? Proteomics. Now, this much indication is enough for biologists to kill the or make the project work. Okay. That is the proteomics played the role. Now, you do whole analysis, cluster, networking, all you have learned in workshop, must do. This is very useful. When you do, you saw tumor suppression protein TUSC3 is getting upregulated. That is the logic. Na? Suppression protein should go up, cancer related proteins should go down. But we found this transcription factor and caspase 14 came up. So, that we do not have anything. Okay? Clue about that. You quickly look, this compound is binding with DNA or not? Control take ethidium bromide and just you look, it is not binding. Quickly next experiment, TM. Oh, TM is changing somehow. No need to find quickly mechanism. Okay? One by one, one by one. Third, you are in CCMB, all are well trained here. Okay? Now, third, quickly look, does E4 after alter cell cycle? Here we found a little bit very different and strange result. You see this sub-G1 population is getting very high with the concentration, dose dependency. Okay, that means you change the dose, IC50 is 18, if you increase this dose, and here you find the mechanism, subG1 population is getting very high, here you see in same thing in 24 and 48 hour, result is wonderful. Something confusion is there. This pattern is same, this pattern is changing in the S phase. No answer we have. Okay, this happens, that means different dose is working at different time point at different things. Okay, but this gives subG1 population is altering the circle. So what is the conclusion? E4 perturbs the cell cycle and cause the cell death. If this we know, then next what we should do? Quickly do apoptosis, and quickly we are showing again with the concentration, increasing the concentration, what you are doing? You are able to go to late apoptosis more. So this is working in the time and dose dependent manner. That's we come. You can't conclude anything more than this. Now E4 cause cell death by apoptosis. Now you go another experiment, DNA fragmentation, run the gel, quickly do it, or do tunnel assay, little bit fancy, and you can see that DNA fragmentation is also going up by higher concentration. So we are trying to find the mode of action, mode of mechanism, and question answer is here. E4 cause apoptosis by DNA fragmentation in dose and time dependent manner, even not binding with the that. So now detection of intracellular ROS, we have saw mitochondrial thing, we have saw ROS protein. So with this we found that this is also making sense, and mitochondrial membrane potential assay we have done. With this we came to that it is something is happening, we are not sure. Okay, something is happening towards the mitochondrial cell. So compound E4 induce ROS generation mechanism and leads to mitochondrial membrane depolarization. This indicates that. Okay. So what proteomics told mitochondria, okay, result came by other biological technique, same or different? That's we need. So with this, now no one is going to believe you cell culture, huh? Only publication. Okay. If you want to move, mouse experiment you have to do. So this is xenograft mouse, you take the cell line, inject in the mouse, make the tumor, leave a month time to stabilize, inject the compound, and if it reduce, supervisor will move next stage and the student will go happy another year for move. Okay. So why this happens? What is the most important things? If you take control, because after 1200 ethical doesn't permit you to keep the tumor size, and if you give treatment, it is completely demolished. This results gives very nice. Even I like this is first experiment. You see, because experiment you have to plan very well. Give injection only on Monday, a student will not feel bad. Monday come, inject nicely, wait for next Monday, and you see the once how dose dependency go. So this proves clear that don't give too much pressure, then they will not think. Okay. So this gives clear that how things are working. If it is really working, what is the next stage? Again use your proteomics. Now we are sitting here, we have this tools, technique, knowledge. We took the tumor from both after treatment, that do full proteomics, do histological staining, quickly show where is the apoptosis is happening, and histology of tumor, thus you can see treatment, yeah, just this. And now I told you one word, side effect. There should not be a side effect. Okay. So now when histology of liver we do, we don't see any change, only compound when you inject. When tumor formed, any compound injected, we don't see in histology any change, and no mouse has died. That is the biggest with this. With this, you see similar thing in spleen, you saw similar thing in lung, heart and kidney. That gives the, now here 2.5 years after hard work of system and paperwork, finally we got knockout mouse retinoblastoma, where gene is knockout, RB minus, P53 minus, so mouse will get automatically retinoblastoma. Here we have started treating and we are getting potential very good result, and I will show you most probably next time whenever we meet. With this I showed you how things will go. Similar concept we have used for leukemia, very nice IC50, 1 minute I will show you the concept is repeating, that you got late apoptosis. What is the interesting thing here got? When you injected, this is another compound, but similar line, in 5 minutes it reach after injection to brain, and if you leave for 45 days, this compound get from body out also. So this is another compound working for leukemia, this is very nice. Third is melanoma, all you know, what I saw how to come the mechanism. This is the last. Here there is no cluster. If it is working, there is no cluster, and when cluster is there, still compound is working, you find mode of action and mechanism, which signaling pathway, very simple. All the known target drug you take, make the slide, and when you find this is the target, and then you do all your experiment, and you will find by antibody this is happening. Okay. I told you in biology antibody is very important. Now you see BCL2 is the down regulated, PAKT is down regulated, and if this is really apoptosis, all caspases should be down regulated or up regulated? Up regulated. Biology is like this. Okay? Only mass spec is not enough. You need mass spec and parallel this. When you reach this, by this anyone can tell there is the apoptosis, caspase is up, there is something is going. If you have this data, you make your models how mechanism is working. When you go to this mechanism, we saw caspase 8, 3, 7, polyadipure ribogylation, caspase 9, FF, cyto-C, BCL2, BAX. So this is the mode of that, and mostly it is going towards the AKT signaling pathway. Okay? Now use your knowledge, here experience is experience. Okay? Then you see it is AKT, FOXO1, BCL2, P53, BAX, apoptosis. This is the chain, it is coming down regulated and up regulated. So finally we are coming, it is towards the AKT signaling pathway. With this, thank you all of you, your patience and for time.
From today's lecture by Dr. Suman Thakur, I hope you got a complete image, understanding about why getting a single biomarker for cancer is tough, why many biomarkers have not been able to reach to the translational work so far in the clinics. Dr. Suman Thakur showed you how multiple cancer cell lines having same protein but different amount. This is the reason a labeling technique like SILAC or iTrack can be helpful. He also showed a complete overview how cell biology can be used to get the drug screening strategy, and animal model experiment can help in validation. I hope you are gathering different facts from different scientists, clinicians, and trying to understand that what are the latest advancement in the field, and also what are the gaps from the clinical as well as its successful translation, in which way by integration of cell biology, various type of omic based technologies and clinical strategies together, only we can make some success in this area. The next lecture is by Dr. David Fanyu, who will talk about predictive analysis. Thank you.